Hi, everybody. If we're all ready, we can start. I'll wear my glasses because it's a little dark. So good afternoon, everybody. My name is Jacqueline Horsington. I'm an Australian working for EUFMD. And I'd like to welcome you to this session called Fast Vaccine Innovations and Selection. So FMD has a very long history. I actually read for the first time the other day that the earliest description of probable foot and mouth disease was back in 1514 by a monk living in Venice, which I thought was pretty snazzy. And the first experimental FMD vaccine was produced almost 100 years ago. And while methodologies have changed and improved, the most widely used vaccines for FMD are still inactivated virus vaccines. And also, while many high-quality vaccines are available, poor quality vaccines with low efficacy or limited stability are still widely used across the globe. So in order to break established paradigms and have tangible positive impacts, innovation is essential, not only in vaccines themselves, but in the approaches for assuring their quality and for optimizing their use. So what's in store for you in this late afternoon session? I hope you've all had lots of coffee. The first half focuses on new vaccine technologies, reporting on some of the latest developments that could influence future vaccines for FMD and other fast diseases. And the second half will address issues related to vaccine quality. We're going to have a few speakers, and then they'll come up for a Q&A at the end of each of the two halves. And I'll ask my speakers to stick to time so we can make the most of the Q&A time. Before we begin, I have to give a small disclaimer for the first 4 presentations. Just so that you all can note that these studies include the use of a commercial vaccine, and the research received funding and support from a commercial company. So to begin this session, I'd like to introduce a colleague and friend from one of the countries I call home. Please welcome Erwin van den Born from MSD Animal Health in the Netherlands. Erwin trained as a virologist and molecular biologist in the lovely cities of Wageningen and Leiden, as well as completing a postdoctoral fellowship in Norway. He's currently a senior R&D project leader at MSD Animal Health in Boxmeer, developing vaccines against a number of viral diseases in pigs. And today he'll be talking about a new vaccine for our favourite virus, foot and mouth disease. Erwin.
Thank you, Jacqueline. I want to talk to you about serotype A, about G-VII. And so I go to the next slide. The AG7 lineage was around for quite some time, but in 2015 it was hitting hard, and we found out that vaccines were not, the available vaccines were not protecting well. However, after a while in the scientific literature, it was found that if you have a high potency vaccine based on another A serotype, you can get some cross-reactivity against this AG7 lineage. However, if you want to have very good protection, you need an AG7 strain in a vaccine to get this good protection. So the aim of our study was to develop a vaccine based on virus-like particles based on an AG7 lineage strain. So we produced the virus-like particles in the baculovirus expression system. And in order to produce them, you have to stabilize them, because we know that these virus capsids are very unstable. And without mutations in the virus capsid proteins, you cannot keep them intact. And that is especially important for thermostability, as you can see in the graph on the right-hand side, that if you heat treat those virus-like particles for 20 minutes at 56 degrees, you can keep the stabilized capsids largely intact, while the wild-type capsids completely fall apart. The first study we did with these virus-like particles is a dose-finding study. We wanted to know what type of dose we need to get sufficient protection. And I know that there's a lot of variation in those type of animal trials, and that the steps between the doses are not so big. But we look at the bigger picture. So protection is one thing, but we also look at the height of the virus neutralizing titer. We look at rectal temperatures. We look at viremia after challenge. And looking at this, I think we were aiming quite well with our normal dose, what we call the normal dose. That one gives the best characteristics that we need to move further. So the further studies we want to do is to see what the protective capacity of the virus-like particles are in the different species, sheep, goats, cattle, and pigs. And we also, of course, want to determine the PD50 value of such a VLP vaccine. Before we do so, we have done already a couple of other studies. We created multivalent VLP vaccines, and we looked at cross-reactivity. And as you can see in the table, that after prime boost, we get really good homologous titers, but also good cross-reactive titers. It's assumed that titers above 1.5 are predictive for protection. So we assume that this VLP vaccine could protect animals against the strains listed here in the table. We also looked at duration of immunity, and we have seen that especially if you increase the period between prime and boost from 1 month to 3 months, you can get a duration of immunity of 12 months with this multivalent vaccine, the same multivalent vaccine as we have used in the cross-reactivity study. You have heard us talking about virus-like particles, maybe for the last decade or so. We in MSD, but also our collaborators at Pirbright Institute, University of Oxford and University of Reading. And I just want to highlight what it takes to bring such a product to the market. I get this question quite often, why does it take so long? And I just want to highlight that there are a couple of phases in product development. The research phase, then the development phase, registration phase, and then market authorization phase. And every phase takes some time, and in animal health things go faster than in human health. For sure, because we can test vaccines right into the animal model. But we, with the VLP project, have been in this first phase for so long. And the reason is you have to tackle with this new technology so many aspects. You have to look at yields of virus-like particles. You have to look at the different disease models for the different strains that you pick. Just picking strains will take also a lot of time because you want to pick strains that cross-react well. We also have been developing an in vitro potency test so we can release batches not with cattle anymore, but for animal welfare reasons with a simple lab test. We also have been looking into adjuvants. And all these things have to be tested in animals, and usually this has to be done in high containment facilities. So once you have crossed this step, you go to development. Again, for every strain, for every species, you have to show that the VLPs are working, for instance, in PD50 studies. And so if you have 4 species and, say, 10 different strains, you have 40 different high containment animal trials that you have to perform. You can perform them at once, it's subsequential, and costs a lot of time and money. So we are somewhere in this process, and we're making good progress, but just to highlight that for a new technology, you have to tick all the boxes and you have to go through all the steps. So back to AG-7. I hope I could convince you that we have produced a virus-like particle vaccine against AG-7, and that this vaccine can give protection in an homologous challenge, but also could protect in a heterologous way, and that we can get a good duration of immunity. And with that, I would like to thank you all.
Thanks so much, Erwin. Okay, next I'd like to welcome Eva Perez-Martin. Eva is a veterinarian and research scientist. She completed her vet degree and PhD in Barcelona before moving to the US to work on biotherapeutics to control FMD, working at Plum Island. She then returned to this side of the Atlantic and currently works at the Pirbright Institute, focused on developing better FMD vaccines for cattle and understanding FMDV transmission in African buffalo. Thanks, Eva.
Hello, everyone. So yeah, today I'm going to talk about the VLP vaccines that we did focused on Eastern Africa VLP vaccines. So why we decided to study a new platform for FMD vaccines when the kill vaccines really work very well? Well, we think that the VLP platform offers several advantages that the kill vaccines don't have. Like for instance, that there's no live virus required, so they are safe. No virus escape. There's no importation of viruses to endemic countries. You don't need high containment facilities in order to produce the vaccines, which are very expensive to build and even more to maintain. So you can enhance as well the production of the vaccines because you can produce them in any BSL-2 lab. So in case of stable VLP vaccines, we know that they are less reliant on cold chain, very important for transporting. They are also DIVA vaccines, very important fact. They are very rapid to generate, so in case on an outbreak in an endemic country, it's very easy to sequence that virus and generate the different cassette to express the VLPs in the baculovirus system. And also as from the research point of view, it allows us, now that we know very well how this VLP works, it allows us to tag the VLPs with different epitopes and drive those VLPs wherever we want in the immune system. So these are the advantages of the VLP FMD vaccines. However, this project also came with different challenges that we had to overcome, and 3 of them are very important. The instability of the VLPs, the poor yields that we can produce in the baculovirus system, and the third, that we needed a method to quantify the VLPs, since we couldn't measure the RNA from like we would do in a kill vaccine. So with regards to the first challenge, So we knew that we had to insert some mutations in the capsid of the VLPs, and for that we use an in silico modeling that it's able to predict the 3D structure of a sequence given. So with that, we were able to identify those amino acid candidates of being mutated in order to confer stability to the capsid. So here we can see how, for instance, in 2 adjacent VP2 proteins, if we change the amino acid for a cysteine, the histidine for a cysteine, we were able to create a covalent bridge there that was very stable. And this is a video that I could borrow from my boss just to show different amino acids in between 2 VP2. The second challenge was to increase the yields that we were obtaining in the baculovirus system, and for that we followed 2 different strategies of the P12A3B precursor that we were using. So as you know, the P12A3B is the precursor that we used, so that also with the 3D that will cleaves itself and to then cleave the structural proteins from the P1. Well, the 3C protease is very toxic to the mammalian cells, but even more to the baculovirus cells. So that's why we had to follow 2 different strategies in order to reduce the activity of the protease. The first strategy was to do a site direct mutation in the sequence, as you can see there. And the second strategy was to introduce HIV ribosomal framesheet also in the sequence, so that the combination of both the strategies, because with the framesheet, what we were doing is to reduce the expression of the 3C protease. So it's the combination of these both strategies what allowed us to really express the VLPs without being toxic to the cells. And the third challenge was to quantify the VLPs and also the stability of these VLPs. And for that, we had to develop some in vitro test that we called integrin or DAS ELISA test, which are able to discriminate the intact VLPs from the 12S. So for that, we use antibodies that are specific for the intact capsids or the 12S, antibodies that can be LAMA antibodies already described by the Harmsen team, We also use some monoclonal antibodies developed by MSD or some bovine recombinant antibodies developed at Perrite. But what we know is that they are specific for the intact capsid or the 12S capsid. The tests that we use for the VLPs are serotype specific tests, but at Perrite we are also developing another universal test, and I think that Stephen Berryman will talk about it later, in order to also quantify the killed vaccines. So as you can see here in the graph on the right, we could perform some thermostability studies in order to see how stable our mutants were, as well as quantify the amount of protein we were working with. So now that we are able to produce VLP vaccines, we wanted to test the immunogenicity of these VLPs. So for that, we expressed different VLPs, Otter, O Ethiopia, Egypt, A Kenya, and 2 SAT2s and 1 SAT1 Kenya, with different mutations. Some of them with a single mutation, some of them require a double mutation in VP2, some of them require a double mutation in VP1 and VP4, and some of them 6 mutations, like in the SAT1. That's depending on the serotype. The mutations that we have to perform are different. So we vaccinated the 5 animals per group on day 0, and then we boost them at day 28. We performed the homologous VNTs to see the dynamics of the antibodies, and also the cross VNTs to see if we could use these vaccines in Africa, according to the AG results. So here I'm showing you the dynamics of the antibodies at different days post-vaccinations, and as you can see, all these vaccines were able to induce high titers of antibodies as soon as 7 days post-vaccination. So even for the SAT2, which is a difficult serotype to work with, the antibodies were pretty decent. So we were very happy about these results. So we took day 21 and day 35 serum samples from these animals, and we decided to test them against the panel of viruses that the AG results indicates in order to use a vaccine in Eastern Africa. So, and they have a criteria for a pass, meaning that 3 out of 5 animals with antibody levels above 1.5 log10, for 3 out of 4 isolates in serum from day 21 post-vaccination or 10 days after the boost was a pass. So we tested the all Ethiopia VLPs, and they were a pass even after first vaccination. The SAT1 Kenya VLPs as well, with the Egypt as well. However, the A Kenya didn't pass. They didn't pass after 1 vaccination, but the antibody titers were high enough after the boost. And then with the SAT2 vaccines, both of them were a pass. We decided to include as well the SAT2 topotype 14, the one that is causing outbreaks in the Western Asia nowadays, or 2 years ago, and it's also protected. The SAT2 vaccines also protect against these isolates. So we were very happy about the results. So here I'm showing you the monovalent vaccines for Eastern Africa. However, we know that from another study that we performed, where we had animals vaccinated with a tetravalent vaccine and also a pentavalent vaccines, as I'm showing you here, with different Asian strains and also African strains, Saudi, Asia Shamir, Kenya, or Turkey, and SAT2 Sudan, in case of the pentavalent vaccine. And we can see how the dynamics in these polyvalent vaccines is high as soon as 7 days post-vaccination, and remains high against all the viruses tested, the homologous viruses tested, all along the experiment. So just to conclude, I hope I convinced you that the VLP's vaccines show great potential as a tool in the fight against FMD in Eastern Africa. And if you want to know a little bit more, Ranjita Bomana, my colleague, is going to be with a poster where she used also a tetravalent vaccine using the VLPs. And finally, to acknowledge all the people involved in this project, which are many of them, especially University of Oxford, University of Reading, MSD, and the funders, Bill and Melinda Gates Foundation, and the Wellcome Trust. And thank you all.
Thanks, Eva. Okay, our next speaker is Justine McPartlan. Justine's research career has focused on novel RNA vaccine strategies and the development of a nanoparticle delivery platform, as well as applying CRISPR technology to cancer research. She currently works as an R&D lead and project manager at Teva Biotech in the United States, and is talking to us today about the development of mRNA vaccines, including for FMD, for use in livestock. Thanks, Justine.
Hi, I'm really excited to be here representing my company, Teva Biotech, and presenting our work on livestock vaccines with a focus on foot and mouth disease. And I do have a slight history of fainting, so I decided to sit for everybody's benefit and my own. So Teva Biotech is a biotech company located in Boston, Massachusetts in the US, and we're focused on developing a library of delivery materials for RNA vaccines and therapeutics. We spun out of MIT over 6 years ago at this point, and since then we've been further fleshing out this library of materials. So we have a patent-protected library with some materials for use in vaccine applications and other vaccines for use, or others for use in therapeutic applications. We first focused on human vaccines, and we've been very fortunate to receive funding from the US government and from CEPI to develop vaccines against influenza, COVID-19, Japanese encephalitis virus, and tick-borne diseases as well. We are now working with New South Wales, the government in Australia, to develop a livestock vaccine program. So our technology is the RNable platform, and RNable ABL is aminoalkyl branched lipidoids. So we're developing this diverse library of ionizable delivery materials, which are separate from the cationic delivery materials and the polymers that are traditionally used for mRNA delivery. This is an alternative to conventional lipid nanoparticles, and in the development of these materials, we focus on safety and biodegradability. So we're very fortunate to have received funding from New South Wales in Australia, and we have a consortium of partners that we're working with, including the Department of Primary Industries, the Elizabeth MacArthur Agricultural Institute, the Canadian Food Inspection Agency, and the UNSW RNA Institute. And the focus of this collaboration is to protect the agricultural sector from emerging infectious diseases with mRNA vaccines, and also to develop a domestic mRNA manufacturing facility in Australia. So our FMDV vaccine strategy is utilizing mRNA, which is a little bit different than some of the other strategies that we've seen today. So our antigen of interest is the P1 capsid polyprotein, and we also want to include the 3C protease for proper processing and presentation of the viral proteins within the P1 polyprotein. So our strategy is to express the 3C protease in trans from the P1 polyprotein. So we have one molecule encoding the P1 polyprotein and another RNA molecule encoding the 3C protease. What we can do is we can co-formulate these mRNA molecules at a fixed ratio, whether that be a mass ratio or whatever. So we can co-formulate those and introduce both the P1 and the 3C into the same cell, so you have proper processing and presentation of those viral proteins. So we determined an initial panel of RNable delivery materials through an internal screening process, and we're using N1-methyl pseudouridine modified mRNA for these studies. So this also allows for DIVA capability because we are not including all of the proteins from FMD. So to start here, we did an initial study in mice. We work with mice at our facility, and our partners are doing larger animal studies. But mice were vaccinated with RNable candidates. There are two here that were included in this initial study. And these two candidate delivery materials were formulated with 4 different conditions. One with just the polyprotein, the P1, which is represented by the 0 here for each set. And then the other formulations were all the P1 polyprotein with the 3C in trans at different ratios, where we were titrating in the 3C, attempting to minimize any toxicity from the TC, but also make sure that we're getting proper processing. So here we have ELISA results. This is a competitive ELISA. It's the PrioCheck type O antibody kit, and our vaccine is based off of type O. And you can see here that based on the results of the kit that we were getting greater than 50% inhibition with RNA-BOLT2. So we were really excited about this, and we saw, we saw an improvement in vaccine efficacy with inclusion of the 3C beyond just the P1. So we decided to proceed with our partners at EMAI in Australia, and we took these vaccine candidates, and we further expanded the panel to include other materials that we had developed between the first and second study. And we tested a variety of ratios of P1 to 3C, as well as a panel of different materials. And we found that with our lead material and the best performing material with the best ratio, we were able to get a strong FMDV neutralizing response in all rabbits that were vaccinated. And this work was done at CFIA, where the VNTs were completed. We then decided that we wanted to move forward into cattle. It's important to test in the target species, as we noticed the difference, even just between mice and rabbits. There's a difference in which ratio was the best performing. So we were really excited about this data. This data is from an ID vet FMD type O competition ELISA, and we vaccinated cattle with a prime boost regimen, 50 microgram doses, and a 4-week interdose interval. And this was a serum sample that was taken at 2 weeks post-boost. So we see here that based on the kit, we were getting above 90% inhibition as a result. So antibodies were present in cattle from all of these vaccinated groups, and there were 6 different candidate formulations. So to get a little bit more resolution, we were able to work with FLI to test these by VNT, and we were really excited about the results that we had here. In these cattle that were treated in the same regimen as the previous slide, we saw that we were getting robust seroconversion observed for several of our candidates, and we also identified a lead candidate, candidate 2 here, that we are hoping to proceed through challenge studies with. So in conclusion, an mRNA vaccine strategy has been identified for foot and mouth disease, and the delivery chemistry does clearly impact the efficacy of the vaccine candidates. So with the 50 microgram dose that we used in cattle, we do feel that there is room for improvement. We know cost is a major factor for livestock vaccines, so we are hoping to decrease cost, potentially proceed with a prime-only vaccination regimen, and also test out a shorter interdose interval just to make this more realistic for livestock. So we are proceeding with a challenge study. Up next, we have that scheduled, and we're also hoping that we can utilize some of the advantages of mRNA vaccination by testing out different strains and adapting rapidly to different strains of interest. So we're seeking partners interested in virus-free FMDV vaccine production, and I'm happy to talk more with anybody who's interested about our vaccine platform. So I'd like to acknowledge all of our partners. Shawn Babiuk is here. He's going to be presenting next, but we have been very fortunate to partner with Australia and Canada to work on these vaccine platforms. Thank you. Thank you.
Thanks so much, Justine. That's great. Okay, our final speaker in the first half of this session, joining us from a city very dear to me, not least because of their amazing ice hockey team, is Shawn Babiuk from the Canadian Food Inspection Agency in Winnipeg, Canada. Shawn completed his undergraduate and postgraduate degrees in Saskatchewan, followed by a postdoc in vaccine development. He then moved provinces, working for the last 10 years at the CFIA in Winnipeg. As well as being an adjunct assistant professor at the University of Manitoba, he is currently the head of the Vesicular Diseases Unit, focused on the development of diagnostics and vaccines for several foreign animal diseases. And today he's presenting on a new technology vaccine for lumpy skin disease. Thanks, Shawn.
Excellent, thank you. So we're going to talk about development of mRNA vaccine for lumpy skin disease. So this is work in partnership with TIBA. So again, they have all the expertise in the delivery of mRNA. Again, we didn't have that technology, so it was better to partner with a company that had that. And then we also are working closely with Australia, both CSRO as well as EMAI. And this really came from conversations about the risk of lumpy skin disease entering Australia. And I said, well, why don't you try and make a mRNA vaccine for lumpy skin disease? So again, we were successful in convincing our stakeholders that that was an important issue, so we will go on to the reasons. So we know lumpy skin disease continues to spread into new regions, very difficult to control. And again, there's existing attenuated lumpy skin disease vaccines. They're effective in assisting control efforts to eradicate the disease, but they have limitation. One being the inability to differentiate infected from vaccinated animals. And this limitation and others prevents the preemptive use of vaccination in countries that are at high risk for lumpy skin disease outbreaks. And there's been work demonstrated that inactivated lumpy skin disease vaccines have been demonstrated to be effective against experimental infections. So this proves you can make, vaccines to lumpy skin disease. The big question is, what are the protective antigens? And we don't know that. So to make a long story short, we could talk about the protective antigens for the whole entire hour or days, but we have 156 potential antigens. So we went and knocked that down to look at, what are the potential antigens could be involved in neutralization. So we go and knock that down to the 30 potential antigens, which are membrane proteins. Then we can further identify antigens that are immunogenic. So which of these membrane antigens following infection generate antibodies. And then we can also look at the research. So again, not much research done on lumpy skin disease, but we can look at other pox viruses to see homologs, and then we can identify the antigens and then test them in vitro and in vivo. So we identified 6 different antigens, and our first study was in mice. So we went and ran mRNA vaccines with single antigens, and then looked at the immunogenicity in mice and rabbits. So in mice, we see several antigens giving virus neutralization titers following immunization, and also in rabbits, although the mice seem to give higher neutralizing responses. So next we wanted to do the studies in sheep. So we have 6 different antigens and then combinations of all the antigens in sheep. So what we can see is that some antigens gave some neutralization following the boost, and then duration was also looked at, so another 2 weeks following the second immunization. One of the animals with a single antigen gave some neutralizing antibodies. Now what's interesting is if we look at all the combination of all the antigens, we can see we can get neutralizing antibodies at after the boost, and then these antibodies have a longer duration. So we really wanted to look at, does the combination of sera from these individual antigens play a role in immunization? So we went and took the individual sera from all the individual animals, and then we did the mixture of seras and looked at the virus neutralization. And what we can see from these results, if we use 2 sera combinations, we can see that different antigens together give us neutralizing antibodies. And then if we go to 3 sera combinations, we get more neutralizing antibodies with the combination, and similar with 4, and then 5 sera combinations, we get improved neutralizing responses. So this really shows that multiple antigens are important, or very likely important for immunity against capripox viruses. So then we wanted to look at cattle. So we did a dose experiment using a single antigen at different doses, and we can see that the single antigen with different doses, although all the responses are detectable by ELISAs, again, we had some neutralization levels. And then if we took 4 different antigens with a lower dose of antigen, or lower dose of mRNA, we can see that those gave better neutralizing antibody responses, as well as antibody responses to the individual antigens by ELISA. So we can mix the mRNAs together with multiple antigens and get synergistic effects of the neutralizing antibodies. So the conclusion, so we have our mRNA vaccines. They're immunogenic in sheep and goats, or sheep and cattle. So again, we could also use this for sheep and goat pox vaccines. Some antigens induce neutralizing antibodies. Combinations of sera elicited from single antigen mRNA vaccines improve virus neutralization. And mRNA vaccines using combination of antigens induces stronger neutralizing antibodies with a longer duration. So we have to do the challenge studies in sheep as well as cattle to actually determine the efficacy of these vaccines. And again, these mRNA vaccines can allow DIVA once the companion diagnostics have been developed and validated. So again, thank you.
Thanks, Shawn, and thanks to all the speakers for being good on time. I'd like to invite the first 4 speakers to the stage now, and we can do some questions. Shove over. Okay. Thanks, guys. We might start with Alejandro. Do you have a mic?
Thank all of you for a fantastic session, very nice talks. This is to Justine. Is it really cost effective to produce 2 RNA molecules for a single vaccine for cattle? Or you did it because, are there any size restrictions of what you can put on the same molecule? So why taking that alternative? Because it doesn't seem like it will be really cost effective to produce 2.
So the question is about why are we introducing it as 2 molecules rather than cis? Okay, so yeah, the reason that we decided to take this approach, it was something that we had experimentally determined was the better approach. We wanted to be able to titrate in the amount of 3C protease and really control the amount of 3C protease in relation to the amount of the P1 polyprotein. So cost-effective wise, I think that we've seen that there have been decreases in cost of production for DNA over time, and we're hoping that we'll see the same thing with RNA as well, as more people and more companies are investigating RNA as potential drug product. So we're hoping that costs go down, and if we can minimize the dose that we're utilizing, if we can minimize the number of administrations needed, we think we can decrease cost in that way as well. So we did decide that the 2 mRNA molecules as separate molecules would be better as a product and would be more effective, and we're hoping that in doing that we can minimize those other factors.
Okay, and I have another one. It's okay. For the people working on VLPs, the payload of the VLPs that you are using for the animals are within the same range that you use the whole viral particle? Or I'm not asking the exact payload, perhaps you cannot tell me, but are they within the same range? I'll leave that to you.
Thank you. It's exactly in the same range.
Exactly on the same range.
Yeah, we have seen that the virus-like particles behave exactly in the same way as inactivated virus. So in terms of payload, onset of immunity, duration, and we use the same payload, and it works well. Yeah.
And do they work in one dose, most of them, or you always have to give 2 doses? Because of course you are lacking the RNA that activates TLR7 and all the story, but just asking. You always need 2 doses, or?
No, well, standard tests, PD50 tests are always with 1 dose and then challenged 3 weeks later, and that works well. But in naive animals in the field, you have, even with inactivated vaccines, you have to do prime boost to get usually a good response, and that's not different with the virus-like particles.
Thanks, Alejandro. I might go Elizabeth, Aldo, Danny. Sorry if I got the order wrong, but...
This is a question for the VLP, for Erwin. You mentioned the need to include stabilizing mutations on the serotype A, and relatively speaking, the serotype A is one of the more stable in the natural occurring viruses. is the need of this additional mutation being introduced for ensuring protective immunity being induced by the VLPs, or you also are looking for increasing the shelf life of the product in the long-term storage?
Yeah, thank you for the question. As you could see in the graph with the heat treatment, that wild-type capsids just fall apart. They don't have the RNA inside that normally functions as a glue to keep the structural proteins together. So with this glue lacking, we need to have stabilizing mutations, even in stable strains like coming from the serotype A. So for just keeping them intact, and especially for having a good shelf life, you need to stabilize them. And even within the A serotype, there are strains that are less stable. And so we have seen differences between the different A strains in terms of stability.
Thanks, Owen. Aldo?
Yeah, I saw in in almost all the presentations that prime boost is standard, and for foot-and-mouth disease vaccines, it's clear. It's one shot should protect, and also should protect for 1 year. So I'm a little bit worried that we're overestimating the effect of these vaccines, and especially if you go back into literature where in the Chong and Dole, or Dole and Chong, the potency tests in guinea pigs, by the way, which is not optimal, but they show that you need 12 times as much 75S compared to 146S. So I'm still, do you really think that a prime boost strategy is commercially really an interesting option?
I guess this question is for me.
You're too popular, Owen. That's all right.
Yeah, you know, I can only speak from the experience I have with virus-like particles. I'm not up to speed with all classic vaccines, to be honest. But I think if you really want to have a powerful duration of immunity, you have to have prime boost. And that's I think, with one shot, I don't think you manage to get 1 year in naive animals, honestly. But maybe I'm wrong, but this is my observation in terms of serology.
You guys can argue about that later, perhaps. Danny?
Yeah, we can argue about it.
Yeah. My question is also on the VLPs, and more specifically on the in vitro potency test that you are targeting. I fully understand, of course, the benefits of an in vitro potency test over an in vivo one, but the difficulty is often to get it accepted by the regulatory authorities, because if your test is only actually determining the antigen or the quality of the antigen, the integrity, you also have the adjuvant part. So my question is, do you also have a test that would reflect the quality of your adjuvant, or that somehow will also say something about the interaction or the immune response, because that is actually the most critical part with respect to licensing.
Yeah, so indeed, in vitro potency tests have benefits, not using animals, faster, cheaper, all sorts of benefits. But the downside of it is that you only look at the antigen and not to the whole vaccine formulation, the whole vaccine matrix. So there are different components in the adjuvant that you cannot test outside an animal model, for instance, in an ELISA. However, MSD and Berryman and many companies here in the audience work according to GMP, meaning that you have, for every component, every active component in a vaccine, you have to have test, laboratory QC tests. So in our laboratories, we test for the adjuvanting components, and they have to have a certain level in this vaccine formulation. And you have to have a test on the antigen level. And those have to be at a certain level, and that gives you assurance that your vaccine will work in the field, even though you haven't tested this in animal during batch release.
Okay, I think the gentleman behind Danny, sorry, I don't know your name. And then Brian, and then Don, yeah. And then we probably run out of time after that.
Thank you. To all the presenters, well done. I've got 3 questions to everybody. Same question.
3 short questions.
3 short questions. First, do we really consider neutralizing antibodies sufficient enough as correlates of protection? Did we, at any point in time, consider breed variation in response to your challenge studies, especially for the vaccine meant for East Africa, for example, where you have different breeds of cattle? Have we tried to consider the impact of MHCs classes 1 and 2 in relation to the host immune responses? Then lastly, to Shawn particularly for this, what impact did the mRNA vaccine had on virus shedding post-challenge? Thank you.
Okay, do we want to start with the first?
So, I can start. I would not say that I have any personal say on the impact of VNTs as far as a correlate of immunity. That is what we have right now for our mRNA vaccines as data. We are hoping to proceed to a challenge study next. We're in the process of planning that. So yeah, I don't personally have much to say about that one. Yeah, and can you repeat your second question? I'm sorry.
The second question was consideration for breed of animal.
Oh, for breed of animal. Yeah, so again, for us, as we are, my company is more on the delivery side of things. We're partnering with experts in FMD. Unfortunately, they couldn't be here this week, but they, they're the ones who are indicating which particular strains we should be using, and they have not indicated anything at this point in our development about taking into consideration MHC or different cattle breeds.
Are you guys adding?
Yes. So I think virus neutralizing antibody titers correlate with protection, and definitely we must show in local breeds that, well, basically that's true for any vaccine. We have to show that it works either in laboratory studies or in field studies.
Yeah, I agree with Erwin. I think the best tool that we have to as a correlate of protection is the VNTs. And yeah, I'm happy on how they work, and it's been many studies and models in order to see, if they correlate with protection, and they do. So yeah, I'm happy with that.
Shawn?
Okay, so I'll ask your question with lumpy skin disease. Neutralizing antibodies could be considered a correlate of protection, although we have some protection in the absence of neutralizing antibodies with live attenuated vaccines. So with the inactivated vaccines, they get neutralizing antibodies and they're protected. So we suspect the mRNAs are a combination because you'll get cell-mediated immunity also with them. We didn't measure that, but if we see antibodies with the mRNA vaccines, that's a very good hope that the animals would be protected. So again, we haven't done the challenge studies, but we would think that these vaccines would behave similarly to a cross between a live attenuated or an inactivated virus, where you get complete protection from challenge, so the animals don't get the secondary skin lesions.
Okay, I think Brian was next.
Shawn, there's been some impressive results with mpox vaccines from RNA vaccines in non-human primates, which is a quite robust model. I mean, are any of the open reading frames they're using in those mpox RNA vaccines homologous to the lumpy skin disease open reading for
Well,
him?
let's just say when we picked these things before we saw those studies, and again, there is some homology. So again, we think that, for pox viruses, again, if, monkeypox or mpox or those other ones, people have been successful with the mRNA, I think we can be successful with the capripox ones also.
And Don.
I think this is the final question for Erwin. So you very nicely in your presentation outlined this pathway from a concept of a vaccine all the way through to a product. Are you able to share with us where you are on the scheme? And I suppose the elephant in the room, when can we expect to see the VLP vaccines taken to market? I mean, not to the month and the day, but the broad idea. Where are we?
Where are we? Well, I cannot disclose this, obviously, because it's just the way it is, unfortunately. But we had a lot of hurdles, honestly, and we had to solve them. And we are now in the stage where we really are making good progress. So this is the only thing I can say. I'm sorry.
I think we have time for one more quick one. Just here, yeah, thank you.
Hello, it is to Eva Perez-Martin. Regarding the modified lipids, what you mentioned, does it improve the stability of mRNA, or how is that?
Yeah, so our delivery materials, we have not done formal stability studies. We have seen with similar materials to those that were used in my presentation, up to 6 months stability at 4 degrees. And with that, we then took those nanoparticles and administered them to mice, and we were able to see similar immune responses as compared to fresh vaccines, because we have seen that colloidal stability with mRNA vaccines is a bit easier to maintain above the function of the RNA and whether there's breakdown in the RNA. So we have seen up to 6 months, and we're continuing out those studies, but we have not completed formal stability studies as of yet.
Okay, thank you so much, and thank you for some really interesting talks. I encourage everyone to come up to the presenters during the rest of the conference and ask lots more pressing questions about these wonderful technologies. And I also want to say, for more discussion, particularly related to regulatory issues about new technology vaccines, make sure you all come to the GFRA session on Thursday, where there's going to be a really interesting discussion, including this topic. Thanks very much, and we'll move on. Thanks, guys.
Thank you.
Okie dokie. So for the second half of this session, I'd like to invite to join me as co-chair an accomplished scientist who really needs little introduction in this audience, I think. She's a brilliant virologist and immunologist working currently as principal researcher at the Argentinian National Research Council, as well as being director for the at the Center for Health Sciences at the Inter-American Open University in Buenos Aires. She's also the current CEO of the GFRA, who is hosting, obviously, our beautiful day on Thursday. So please welcome Alejandra Capozzo.
Thank you so much. Okay. Gracias. So to start this session, I'd like to introduce Wannes Philips. He completed his master in Belgium at the University of Antwerp with focus on molecular and cellular sciences, and has been working on canarypox viruses in CNSARO for the last few years, researching the safety and efficacy of vaccines against sheeppox and lumpy skin disease. He's currently undertaking a PhD focused on pathogenesis of capripox viruses in their natural hosts and immune response against sheeppox virus in sheep. And he is talking today about a vaccine efficacy study against a strain of lumpy skin disease that resulted from the use of poor quality vaccines. So welcome, the floor is yours.
Thank you very much. So good afternoon, everyone. I will be talking about an experiment that we conducted to check whether the currently available homologous live attenuated Neethling-based vaccines offer protection against an infection with the newly emerged recombinant strains. Before I do that, I will give a short introduction about lumpy skin disease, and especially the situation before 2016 and the situation after 2016. So lumpy skin disease is together with sheeppox and goatpox virus a member of the capripox viruses. And all these 3 viruses are highly host-specific with lumpy skin disease, or LSDV, affecting mainly cattle and water buffalo with a morbidity rate of between 5 and 45%. The main clinical sign of lumpy skin disease is the formation of nodules on the skin, like can be seen on the picture over here, but fever, weight loss, reduced milk production, reduced appetite, and other clinical signs are often seen. The main way of transmission, as was discussed already a little bit this morning, is transmission via vectors, and up until 2016, the disease was limited to Africa, the Middle East, Europe, as well as Kazakhstan and Russia. So a lot of diseases, one of the most important control measurements is vaccination. For lumpy skin disease, you have in general 2 main options. So you can go for homologous vaccination, in which you use an LSDV vaccine strain to protect your cattle against LSDV. These vaccines have proven to be very effective, both in the field as well as in a controlled environment, as is described in the study on the screen. But sometimes, of course, side effects are seen. Another option is heterologous vaccination, where you use a sheep or a goatpox virus, so some of the other capripox viruses, to protect cattle against lumpy skin disease. Big advantage is that almost no side effects are seen with these vaccines. But on the other hand, these vaccines have shown in the field to offer less protection, especially for the sheeppox-based vaccines. So that was the situation before 2016. And then in 2017, a lot changed for lumpy skin disease with the detection of vaccine-like strains of lumpy skin disease in Russia, which was at the time very surprising, because in Russia they were using heterologous vaccines, so normally no vaccine-like lumpy skin disease strains could have been present over there. At the same time, they also saw that there were a lot of outbreaks at northern latitudes and during winter, so that these outbreaks might not have been related to vectors. So they did some studies, and we at Cienzano also did some studies, and we indeed proved that these new type of strains could be transmitted in a non-vector-borne way. Of course, we were also wondering where did these new strains originate from, and the attention was quickly drawn towards Kazakhstan, because the first outbreaks were seen close to the border between Russia and Kazakhstan, and in Kazakhstan they were using a homologous vaccine. We did a lot of research and investigated a lot on this vaccine, and we found that in this vaccine there were a lot of strains present. So different vaccine strains of lumpy skin disease, a goatpox strain, but also some recombinant strains between the different vaccine strains. And these recombinant strains were highly similar, or sometimes even identical, to the other recombinant strains that were found in Russia, but also in China, Vietnam, Indonesia, Hong Kong, etc., because of course the disease didn't remain limited to Russia, but spread it all across Asia. So that leads us to our study. We saw that the recombinants were able to spread really quickly, but we didn't know whether the currently available vaccines that were developed against the classical 1.1 or 1.2 strain would also offer protection against these new recombinant or clade 2 strains. We want to check 3 vaccines, 2 commercial vaccines. From these commercial vaccines, we know that they are safe in vitro, and we also know from previous trial that they are safe in vivo, so they give almost no side effects, and are also effective against an infection with the classical 1.2 strain. Then we also added 1 vaccine that's currently under development. We tested it in the lab, and this was also safe in vitro. In addition, in this trial, we also wanted to check whether our standard challenge model that we used with the clade 1.2 strain would also work with recombinant clade 2.5 strain. So for this challenge model, we challenged the animals intradermally and intravenously. Finally, the setup of the trial on the bottom of the slide. So we have the vaccinated groups, which are 3 groups because of 3 vaccines, and a control group to serve as infection control and to validate our standard challenge model. Animals were brought into the stable. After 7 days of acclimatization, vaccinated animals were vaccinated, followed for 3 weeks to look at the safety of the vaccines, and afterwards all animals, the vaccinated animals and the non-vaccinated control animals, were infected with a 2.5 recombinant strain isolated from an outbreak in Vietnam. Again, animals were followed for 3 weeks, and the efficacy was evaluated. and at the end, all animals were euthanized. So first, some findings after vaccination. After vaccination, we could say that all 3 vaccines were safe. We had limited clinical signs, and we had no viremia of the vaccine strain. In addition, we also had a clear immune response after vaccination, both humoral, so we had seroconversion in all vaccinated animals between 7 and 14 days post-vaccination, as well as cellular, as we saw an interferon gamma response in almost all animals. Then it was time for a challenge, and I will first describe the control animals or the non-vaccinated animals. And I will describe some clinical signs that we've seen. So we saw a prolonged fever of, in a lot of these control animals, in some cases even up to 2 weeks. We saw a strong local reaction, which can be seen on the picture on the left-hand side, on the top of the left-hand side. So you see the local, the inoculation site, the intradermal inoculation site, which was a very big swelling and some crusts that were formed. We also saw a lot of nodules. In 62% of the animals, we had skin nodules on the body of the animal, but also on the nose and on the scrotum, which can be seen on the other pictures. And we also, during autopsy, saw that one animal had even nodules on its lungs. And a wide variety of other clinical signs like nasal discharge, reduced appetite, etc. were also seen. This was in great contrast with the vaccinated animals, where we only saw a fever spike around 7 to 8 days post-infection. And we even didn't saw this fever spike in vaccine C. We saw a limited clinical local reaction, no nodules, and no other clinical signs. So a first indication that this vaccine would also work against the new recombinant strains. This was further confirmed by the viremia and the viral DNA that we found in the organs at autopsy. So for the control animals, we had 62% of the animals with viremia, while none of the vaccinated animals had a viremia. Then we also looked at the organs, and we collected 27 organs upon autopsy. And you can see that for the control animals, the number of LSDV DNA that was found in the organs was higher, or the number of organs where LSDV DNA was found was clearly higher than in the vaccinated animals. Finally, small word on the immune response after challenge. So all of the control animals seroconverted between 7 and 10 days post-infection. They all had the interferon gamma response after challenge. For the vaccinated animals, they already seroconverted post-vaccination, and they remained seroconverted post-challenge. And we also saw an interferon gamma response after the challenge. Then some conclusions. The standard infection model works as expected. We saw results with the clade 2.5 strain that were comparable to results that were obtained earlier with the 1.2 strain. We concluded that we could say that the vaccines are safe as expected because 2 of them were tested earlier and immunogenic, and the vaccines offer protection against a challenge with a virulent 2.5 genotype because we saw no clinical disease, no viremia, and a limited presence of viral DNA in the organs. Of course, we only tested homologous live attenuated vaccines, so for heterologous vaccines and inactivated vaccines remain to be evaluated. And I can only now thank my team at the Unit of Exotic and Vector-Borne Diseases and the people at the animal facilities for making this work possible. And thank you for your attention.
Thank you so much. Okay, our next speaker is Suzanne Kerfua. Here she comes. Suzanne was awarded her PhD from the Nelson Mandela African Institution of Science and Technology in Tanzania, and currently works as a livestock scientist at the National Agricultural Research Organization of Uganda, with particular focus on foot and mouth disease virus. With a significant experience in molecular epidemiology and virology, Susan's work contributes to the understanding and management of infectious diseases in livestock in Uganda. And today she's presenting on the varied efficacy of FMD vaccines. Thanks, Suzanne.
Thank you. Firstly, I count myself honored to be here to present before you. Together with my colleagues, whose names are up there, we are presenting our research findings on the immunogenicity of commercial foot and mouth disease vaccines against East African strains. Vaccination programs are very important because they actually help to reduce viral circulation, they help to reduce the impact of the disease with the ultimate goal of ensuring that a country is free of FMD. In my country, Uganda, The main circulating serotypes are O, SAT1, O, A, SAT1, and SAT2. So we regularly get outbreaks. At least every year there's an outbreak. And actually, for the last 20 years or so, or 30 years, since 1954, when we first discovered the virus, the vaccination programs are very erratic. They are not planned for, they're really haphazard. So it's mainly reactive vaccination compared to having to plan for these vaccination programs. So vaccination has been very expensive to the country. We are losing millions of dollars every year in the purchase of vaccines. And yet there's not been any post-vaccination studies that have been carried out. So this was one of the first studies for which we did a post-vaccination study. So we sought to establish and compare the induction and longevity of antibody titers that were elicited. We also sought to establish if there was a likely protection that would result from the vaccinations. But also we sought to determine if there is any correlation between the SP ELISA results and the VNT results, because this would be helpful, especially for the labs in the very resource-poor settings, to maybe just run a PVM, to establish if actually the vaccines are doing what they're supposed to do. So we had two groups of animals to which we subjected to two different vaccines. So we had vaccine 1 and then we had vaccine 2. For certain reasons, I'm not going to dispel the names of the vaccine producers. So for each group of animals, we divided them further into two groups. One group of animals were vaccinated at day 0 and then at day 180, that is after 6 months. And the other group of animals was vaccinated at day 0, day 28, that's the booster dose that was given, and then at day 180. So for the results, we worked together with the scientists from Pabbright Institute to carry out the VNT testing, whereas the SP ELISA tests were done at my institute, that is the National Agricultural Research Organization. So the VNT was tested against a panel of 8 viruses, which were representative of the diversity, the antigenic diversity of the viruses in East Africa. And for our study, based on several other studies that have been carried out, we considered log titers of about 1.5 and above as protective, as likely protective. So for our results for vaccine 1, we actually see that, or maybe just to let you know, the bold lines are for the animals that received the booster dose at day 28, whereas the thinner lines are for animals that did not receive a booster dose. And we can very well see that, for example, if you look at the serotype A for the different strains, we see that there is quite a good reaction, an immune response, which is quite good and sustained over the 360 days, because this study was about a year long. So we can see that at least the immune response is sustained. That is for A. For O, you can see that for those that did not receive the booster dose, it was quite bad. But still, for those that received the booster dose, it was fairly good. If we look at SAT1 and SAT2 results, you can see very well for SAT1, we get quite good results, and the immune response is sustained at least over the 360 days. But for SAT2, not quite good. If we look at the results for vaccine 2, quite appalling, not very good. This is for almost all the strains that were tested. So it's not very good, especially for the O serotype. And this has a lot of implications because the serotype O is the most widely distributed and is the most common serotype in Uganda. Excuse me. So yes, that's about vaccine 2. Still same story. Excuse me. Looking at SAT1 and SAT2, for vaccine 2, we still get the same story that even there was no significant difference between the animals that received a booster dose and the animals that did not receive a booster dose. So this was quite concerning. The summary of our findings show that the peaks, the VNT log titer peaks average between 1.47 and between a range of 0.6 to about 3.15. And our results showed that the serotype A viruses were better neutralized than the serotype O. Like I said, this has a lot of implications when it comes to foot and mouth disease control in Uganda, given that serotype O is more widely distributed. We also found out that it was for vaccine 1, there were beta titers significantly higher by about 34%, whereas for animals that received the 2-dose primary vaccination regime that were vaccinated, boosted rather at day 28, they had higher titers by about 20%. So Aldo, yes, you may need to give the booster. Yes. Oh, actually, these are water-based. Okay, you ask me later. So these are water-based vaccines that were used, just to give that disclaimer. When we're looking at the correlation between the VNT and the SP ELISA results, we found that the correlation was not very good. Perhaps maybe we did not do the dilutions. We did not do the dilutions over and over. But we realized that for serotype A, there was a higher correlation compared to the other serotypes. So maybe just a summary of these. The overall correlation was about 0.49, and we had this variation of correlation across the serotypes. So the recommendation, I know Aldo is going to say, but yes, we were actually, from this study, we were able to kind of affect policy of foot and mouth disease vaccination in Uganda, because we presented these findings to the government. And lately, from the last outbreaks that we had, the government was like, okay, we really need to get serious about doing vaccinations at least twice a year. So right now, the policy is that farmers need to vaccinate their animals at least twice a year, considering the findings. But also, I think the vaccine producers recommend that this is the way to go. And then also, we would like to advocate for more regular post-vaccination surveys, because this was among the first ones. So we advocate that we should have more of these carried out, because it makes also the vaccine producers accountable, to give us the best vaccines. Otherwise, we were just purchasing vaccines, millions of dollars being spent without actually checking, are these vaccines doing what they're supposed to do? Because we actually had cases of farmers reporting outbreaks after vaccination. Of course, there are several reasons why, but we want to exclude the reason of the vaccines are of poor quality. And the last one is that vaccine companies should be able to share their sera with us to be able to do better PVM studies. So I'd like to very much thank all these institutions that were very helpful in making this study successful. Thank you very much.
Thank you, Susan. So we move to our next speaker, that is Amélie Poulard. After a career beginning in immunological startup companies, Amélie has worked for a number of years at Berryman in the beautiful city of Lyon, France. Initially in equine and companion animal clinical R&D, and more recently in the veterinary public health technical service team. She's an expert in GLP, GCP vaccine efficacy and safety studies, and currently manages clinical trials for FMD and blue-tongue virus vaccines, as well as post-vaccination monitoring. Thank you so much.
Thank you. Dear colleagues, it's an honor to be here and present to you a study we have done to assess the duration of immunity of an early and an aqueous vaccine in cattle. Some published data and traditional belief in the field seem to suggest that olive FMD vaccine should be more efficient and offer longer duration of immunity as compared to aqueous vaccine. This paradigm influences the strategic approach for FMD vaccination in cattle, in ruminants. The problem is there are no data to support this. So we choose to measure it using a comparative DOI design. Duration of immunity induced by FMD vaccine is of paramount importance for any stakeholder involved in FMD prevention. Why? Because a longer duration of immunity means a reduction of frequency of vaccination and everything that comes with it. Animal handling, animal stress, labor, overall cost and risk of outbreak. We did this study to challenge the validity of the challenge that early vaccines induce longer DOI. We compare side by side our two FMD vaccines, which differ only by the nature of their adjuvant. Starting with the methodology. To strictly compare the effect of the adjuvant, we have formulated on the same day an after-vaxxure vaccine, which is adjuvanted with an aluminum hydroxide and saponin, and also an after-pour vaccine, which adjuvanted with a double oil emulsion. Both vaccines were made with exactly the same antigen, this means same payload and same strain. The composition of the vaccine, was composed with 5 strains: 1 Manisa, 1 Panacea 2, I-22 Arak, Azeron Shamir, and Satu Eri 98. The study was performed in France in our clinical test facility. We vaccinated 16 naive girls, and for the primary vaccination, 16 per group, per vaccine, and for the primary vaccination, we have vaccinated 2 doses, 3 weeks apart, and we have sampled the serum on a monthly basis. Around 6 months later, an half of the animal for each group received a booster dose, and the other half were monitored up to 9 months. The serum were tested in our laboratory based in Purbright and Lelystad. Heterologous virus neutralization tests were performed using a field isolate, which were selected based on the epidemiology and availability in the lab on date. The area under the curve were tested individually, were calculated individually over the 6-month period, and the group were compared using a t-test. Now, moving to the result. On the left, you can see the chart showing the neutralizing antibodies level in the 16 cattle over the 6-month period. On the top right, you can see the group of animals receiving a booster dose, and below, the cattle were not boosted. For O Panacea 2 and 10 field isolate, we have the titer, the average titer, go up to 3 logs after primary vaccination. Average titer were around 2 logs after 6 months, and the titer remained stable up to 9 months. We have the booster effect was significant also, since we can observe titer greater than the titer after primary vaccination. And no statistical difference was observed between the 2 adjuvants. For the O India 2001 he isolate, you can see equivalent trends and similar antibodies titer levels. That's the previous one. For A Africa G4, the titer were lower than those observed with O serotype. However, after primary vaccination, for both groups, titer reached 2.5 log, and they slightly dropped to 1.6 after 6 months. But the booster vaccination has a significant effect. Titer increased by around 1 log. And for SAT2-7 Lab12 field isolate, the trend and titer are equivalent with those of A type. Curves loops comparable between both groups. Even so, a statistical difference was observed in favor of after-vaxxure. There are many factors that influence the efficacy and the duration of immunity of FMD vaccines. the vaccine strain, the vaccine matching of the vaccine strain with the field isolate, the antigen quantity, and last but not least, the adjuvant. To be able to strictly assess the effect of the adjuvant, sorry. The vaccine should be compared using the same vaccine strain, the same antigen quantity. The design of this study respects this recommendation, and it concludes there are no difference between the two adjuvants. So when we compare the vaccines, it's important to remember that differences should not be explained by the nature of the adjuvant, but by all the other factors that play a role in the immunogenicity. Through this study, we have demonstrated that aqueous vaccine and OLI vaccine provide similar long-lasting duration of immunity, and the adjuvant is not the key factor to assess the duration of immunity for FMD vaccine. Aqueous vaccines are a valuable alternative to OLI vaccine for ruminants. Indeed, they generally offer a longer shelf life, they don't require a strict intramuscular injection, and have a good safety profile. This study helps to offer multiple options in the vaccination practices for FMD control strategies. Thank you for your attention, and thank you to my team for their work and their support.
Merci, Amélie. Okie dokie. So it's my pleasure to next welcome Stephen Berryman from the Pirbright Institute. Stephen is a senior postdoctoral scientist with comprehensive experience in both fundamental and applied research on foot and mouth disease. Today he's sharing his most recent work on the development of a novel ELISA to determine the integrity of FMD vaccines and vaccine antigen. The floor is yours, Stephen.
Okay, thank you. So as Jacqueline said, I'll be talking about a universal test for intact foot and mouth disease virus vaccine antigen based on detection of VP4. So as I'm sure you're all aware, FMDV vaccines consist of intact whole virus particles, and these virus particles have a positive sense RNA genome surrounded by a proteinaceous capsid. This capsid, this intact virus particles can also be termed 146S antigen. They are relatively unstable, and they, when introduced into animals, obviously they produce a protective response. If you expose them to heat or acid, then they will dissociate into dissociated subunits, also termed as 12S pentamers, which have reduced immunogenicity. An important part of making a successful vaccine then is that you have sufficient 146S content within your vaccine. So the current gold standard to measuring 146S is sucrose density gradient analysis, which is laborious, low throughput, and requires expensive equipment. Our potential solution is we're developing universal pan-serotypic ELISAs to detect 146S and total antigen, which may ultimately be faster, higher throughput, and have lesser requirements for expensive equipment. So the principle behind this is that, firstly, intact antigen contains VP4, whereas the dissociated antigen lacks VP4 protein. Therefore, if you have an ELISA to detect VP4, you can expect to detect intact, but not your dissociated antigen. On the other hand, VP2 is present in both intact and dissociated antigen, so therefore ELISA to detect VP2 can be used to detect total antigen. And both our VP4 and VP2 monoclonals are pan-serotypic, so we'll detect all 7 serotypes. So just quickly to go through the ELISA. So for the VP4 ELISA, to quantify intact antigen, you have an ELISA plate, obviously, coated with a universal capture, which is your integrin. You then block and add your antigen, which will bind to the integrin, whether intact or dissociated. You will then add your VP4 monoclonal, which will bind your intact antigen, but not your dissociated antigen, followed by a secondary with HRP and obviously your substrate. Because then the antibody binds to intact antigen, you see an ELISA signal, whereas if the dissociated antigen, the antibody doesn't bind and you don't see a signal. A companion to this test is our VP2 monoclonal test for total antigen. It's very similar in that you coat your plate in the same way, add your sample, and then a VP2 monoclonal which can bind both intact and dissociated antigen, followed by your secondary and substrate. And for intact antigen, you see an ELISA signal, and for dissociated antigen, you see a higher ELISA signal actually because of greater epitope availability. So in order to measure total antigen, you would deliberately dissociate the antigen in your sample and then measure it in that way. So when we were developing these ELISAs, we used purified 146S particles to look at the ELISAs, firstly with a VP4 monoclonal. And to begin with, what we actually saw was we didn't see much of a signal. So to overcome this problem, what we actually had to do was to add an extra step, which was this chemical treatment step, a cross-linker step, post-integrin binding, but before the addition of the antibody. And if you do this, then you see a signal with a VP4 monoclonal. And what we think is happening here is that when you have an intact virus particle, VP4 is on the inner surface, so it's not available for binding by the antibody. Whereas when you do the chemical treatment, it causes a change in the conformation such that the antibody can bind. It did some gradient analysis to have a further look at this, and so we took purified virus and passed it down a gradient to separate out the 12S pentamers from the 146S virions, fractionated the gradient, and then analyzed by VP4 ELISA either in the absence of this extra treatment step in blue or in the presence of the treatment step in orange. So when you leave out this step, you don't see any peaks in the gradient, whereas if you add the extra cross-linker step, you see the appearance of this peak representing virions. So the extra step allows virions to be detected. So we did some further gradient analysis to look at the specificity of our antibodies. So we first took some purified virus and either left it untreated in blue or acid treated to dissociate in orange, and then passed it down the gradient, fractionated, and then in the first instance detected using the VP2 monoclonal, which can bind both intact and dissociated antigen. And you can see that in the untreated sample, you see mainly a peak for intact virions, indicating that our sample is primarily intact. And when you deliberately dissociate, that virion peak goes away, as you would expect, and you see the appearance of the pentamer peak. If you take those exact same gradient fractions and detect them with the VP4 monoclonal instead of the VP2 monoclonal, then in the untreated sample, you still see your virion peak, which in the acid-dissociated sample, the virion peak disappears, but you no longer see the pentamer peak. And this shows to us that the VP4 monoclonal is specific for intact virions over 12S pentamers. So we've been applying this, doing some work, initial work to apply this to look at vaccines, and this is just 3 example vaccines comparing ELISAs versus density gradients. So vaccine A contains a good quantity of total antigen by VP2 ELISA and also a good quantity of intact antigen by VP4 ELISA. And this corresponds with a decent sized peak for intact antigen within the gradient analysis. Vaccine B contains a similar amount of intact antigen, total antigen, but a smaller amount of intact antigen, much smaller. And correspondingly in the gradient, you see a much smaller peak for intact antigen. And vaccine C didn't seem to contain much antigen at all, and therefore by ELISA, and correspondingly you see no peaks in the gradient. We also compared these vaccines by serology, and so the vaccine A, which contains large amounts of 146S, all 3 components show good VNT titers. For vaccine B, which had the lower amount of 146S, there was good response to the third serotype in the vaccine, but not the other 2. And for vaccine C, which contains not much antigen at all, not surprisingly, you get poor serological responses to all 3 components. Therefore, the ELISA matches the vaccine serology results. To summarize then, we are in the process of developing universal pan-serotypic ELISAs to assess the 146S content and total antigen content of vaccine antigens, both before and after formulation. This could be quicker and higher throughput than current gold standard, which is the sucrose density gradient UV analysis. We have an ongoing Bill and Melinda Gates Foundation funded project to further develop and validate these tests, working with a number of vaccine companies and other institutions. We've developed a prototype kit version of the ELISA with Isla in Italy, and recently transferred that to some vaccine companies for testing and evaluation. This contains all the reagents required for the test, and the box is shown in that picture, and has a simplified workflow whereby the plates are pre-coated with integrin and pre-blocked, and your primary antibodies are directly conjugated to the HRP, so you eliminate the need for the secondary antibody step. Here's some quick sample data from this kit, for firstly with the VB4 ELISA with some vaccine antigen. From left to right, you have an increasing amount of antigen, so for intact antigen you see an increasing signal, and as you would hope for the acid-treated dissociated sample, you don't see that increasing signal because it's not picked up. For the VB2 ELISA, from left to right, as you increase the level of antigen, for intact antigen you see an increase in signal, and for acid-treated sample you see a more rapid increase in signal because the epitope is more available. Lastly, we are in the early stages of developing serotype-specific versions of these tests by replacing integrin as capture with serotype-specific captures, which would allow components of multivalent samples to be assessed individually. Lastly, it just leaves me to thank everyone, all the institutions that have been involved in this project, including Bill and Melinda Gates Foundation for funding, a number of vaccine companies on the right who've been involved at various stages, and ISLA, who have helped us develop the kit, and a number of governmental institutions with India who've been involved in this process. Thank you.
Thank you very much. We move to our final speaker. So the final speaker for this session is David Mackay. David is a qualified veterinary surgeon with a PhD in immunology and has an extensive history in FMD research and vaccine regulation. He was head of the Berryman laboratory when it was known as the Institute for Animal Health, and subsequently worked at the Veterinary Medicines Directorate in the UK, and then lead the Veterinary Medicines Division of the European Medicines Agency. He currently provides independent advice to governmental and not-for-profit organizations in relation to regulation of veterinary medicines, with a particular interest in promoting vaccine security for vaccines against episodic diseases of animals. So, David, thank you very much.
Sorry, just struggling a bit to get me. We're off. Good. Okay, thank you very much. Well, this is the last presentation today, and this reflects some work that Jacqueline Horsington and I have been doing on vaccine quality control at EU FMD. And as you heard from my bio, I work first in research and then in regulation. And so, in the words of Joni Mitchell, I've seen it from both sides now. And we're going to talk about quality of vaccines from both regulatory and scientific aspects. And I've only got 8 minutes. So first of all, I started by saying, how do we modernize the approach? So what do I mean by modern approach to assuring quality? And then I'm going to talk a bit about why it's currently difficult, actually, for users to really rely on existing regulatory approvals to get much idea on either the quality or the characteristics of their vaccine, and why is it that there isn't this kind of information contained on foot and mouth disease vaccines when it is on most other types of vaccines. And a lot of it relates to the issue of effectiveness. How likely is it that your product is going to work? And I'll talk a little bit about the challenges. We've heard some of it today. And then end up with making some concrete suggestions on where we could go with that. So, very quickly, next slide. So what do I mean by modern testing? Well, in fact, you heard that from one of the previous speakers. The old way of really assuring the quality of vaccines was a quick review, followed by lots of testing for every batch that you do, and that was how you assure your quality. Nowadays, it's very much focused on assuring consistency and quality of production through quality assurance of manufacture through GMP and product testing. And then you can really just use testing for targeted risk-based retesting of products to check that the tests done by the manufacturer are reflected when you do them independently. But what you look is compare foot and mouth with almost any other vaccine, and there's a massive amount of retesting done. And when I use the word testing, I mean what's done by the manufacturer, and retesting is what's done by anyone else. So, why is it that you can't, as a user, get hold of this kind of volume of information? And it's really that paradox that in FMD-free countries, which often have very highly developed regulatory systems, there's very rarely foot and mouth disease vaccines licensed there because there isn't a market for them. For the countries where you really are using foot and mouth disease vaccines, often the regulatory authorities aren't sufficiently developed to really produce reliable documentation, and certainly not publish that documentation on how they've evaluated those products. So really it is very much caveat emptor. The person buying the vaccine has to assure themselves both of the quality and characteristics, and they tend to do that by retesting the products. So, and I'm saying that the product literature doesn't really tell you very much about how the product is likely to perform. And why is that? Well, it's largely to do with this homologous heterologous issue, that the only way you can reliably test that a vaccine that you release onto the market is similar to the one on which you did all your authorization studies, is to do all your tests with homologous virus. Whereas, of course, when you're using it in the field, it's a heterologous strain. And unlike for really, and because of the heterogeneity of foot and mouth disease, you can't really rely on those two being closely correlated, whereas for most vaccines you can. And so you have to have both testing for quality and testing for likely performance against field strains. The biggest issue is around efficacy, and it's really, as we've heard, can you use VNT? Can you look at homologous versus heterologous? The answer is it's really challenging. And the further you get to the right of this slide, the less reliable it is to use your in vitro test to correlate with the tests that will have been done to validate your vaccine. So the WER manual says the definitive test for an FMD vaccine is a challenge test in cattle, and then you can look at in vitro tests and correlate those. You can look at serological tests against homologous virus, against heterologous. The further you get to the right, which is what you actually want, because that is testing against field strains, the further you are from the definitive test that was done to validate your vaccine. And therefore, you have to rely on multiple steps of extrapolation. And I'll talk a little bit in the next slides, what does that mean for the reliability that you can place on the results that you get. So, the conclusion is testing for effectiveness is difficult and expensive. And the final point is, we're all trying to drive now to replace in vivo tests with in vitro, but there is a lot of extrapolation that's required. And what I would say is that based on the experience with every other time when you do that, it requires more than just validation and demonstration of equivalence. It requires a complete change in mindset from all of the stakeholders, manufacturers, regulators, and users, that they are happy to accept those tests. And so it's not just as good as actually demonstrating that it's a good test. So in summary, this actually just summarizes all the testing that's actually already done on foot and mouth disease vaccines, normally either before or after they get onto the market, and it's an awful lot. So to me, the question is not what more tests do you want to do, but how can you rely on the tests that are already done, and how can you make them the best possible? And so I've come up with some proposals really for discussion on that, and some of them will be familiar with some of the talks that we heard earlier on. And the first really is that you need to be able to rely on the tests that have already been done. And that really is, you don't just do testing, you look at methods of manufacture, you look at the testing done by the manufacturer, has it been done to the right standard, you look at GMP, and that's really what's called qualification assurance. We seem to be missing the bottom of the slide off there, but never mind. And I would say don't reinvent the wheel. WHO have a very good scheme for doing that, which meets these criteria, which are really the important ones. It needs to be independent, scientifically robust, internationally accepted, does not infringe or impact on the sovereignty of national authorities. That's the really tricky one, and covers documentary compliance, inspection and testing. So, and we at EU FMD, that's what we've been working on this PQ scheme, and if you look on the website, you'll be able to see what we've done about independently assuring the qualification assurance. How much reliability can you place on the existence of documentation that already exists, giving you that? So the more products that go through that, the more users can say, oh good, I don't need to do retesting, I can rely on that documentation because it's been independently validated. Next one. So, and this came up earlier, we really have to encourage, promote, urge, or require, in fact, and if you look in most other vaccines, it's actually requiring manufacturers to supply these reagents to allow laboratories to do the necessary testing independently to assure the quality. And so they do that by making technology transfer of tests a requirement for actually getting a license. It's interesting to say that is the tradition for most vaccines, but it isn't for foot and mouth. Why not? And maybe the manufacturers can discuss. And where you see that these things happen, and South America is a really good example of where this has happened, you get the improvement in the quality of the vaccines, and you get the impact that vaccines have on disease control. So there are suggestions you can, you'd get it for licensing, for pre-qualification. Distinguish what you're testing for. Is it for quality or is it for fitness for purpose? And get manufacturers, as far as possible, and we could talk about this for a long time, where possible, get claims for fitness for purpose, in other words, where it's suited for, onto product literature. Then you've got something to hold against the performance of the vaccine. You can actually rely on the information on where that product is likely to be attested. And again, these are on suggestions on how it's done. And we've heard about the Per-Bright virus panels, which is a very good way of doing it. And then final product batch testing, it doesn't happen that much. And that's really a shame. When we eradicated foot and mouth in Europe, every batch was tested before it went on. Countries that use vaccines really should insist on final product batch testing. And then, oops, that's it. Final. And so in summary, what am I saying? I'm saying really, what I would term modern regulatory best practice isn't routinely applied to foot and mouth vaccines. You need to clarify what is it you're testing. Is it the product, the quality of the vaccine, or its fitness for purpose? And that has implications on the testing you do it. We do need to improve these tests for effectiveness, and that's what we talked about with the various panels and so on. And whilst it's true foot and mouth disease is pretty unique in terms of its antigenic variability, that shouldn't always be used as a reason to treat it as an exception. And the final point, as I say, experience shows that technical improvements alone are rarely sufficient. You really have to get all of the stakeholders into the room and knock heads together to actually get these in vitro and improved tests in place. Thank you. Sorry, I went on a little.
Thanks, David. 8 minutes really isn't very long. I would like to ask all the speakers to join me on stage, me and Alejandra, and two of you please BYO chair. We're going to switch up a little. we good? Thanks so much. Oh, here we go. 5 really interesting presentations. So do we have any questions? Danny, you look like you're first in. So one moment for Nadia.
I have a question on lumpy skin, which is actually for Wannes, but also for David. We all know by now, or we should know by now, about the disaster that this recombinant strains caused due to a contaminated master seed from a local vaccine manufacturer. And my question is actually, how can we prevent this for the future? And shouldn't it be time, let's say, maybe to set something in a regulatory system or in any other way that we could force local manufacturers to make their seeds available for maybe testing in a reference lab to make sure that these type of disasters are not going to happen tomorrow with another virus?
Yeah, I mean, I agree completely, and I think that comes to that issue of actually enabling authorities where the products are used to be able to do batch control and ensure that they know what they're testing for on their batches and that batches are actually tested for, say, extraneous agents. And the second point, I think, would be really ensuring what is, countries, in principle, no country should be using a vaccine that doesn't comply with the WER manual. But what does compliance mean? That's often the real challenge, and I know we've discussed this before. And it says that you shouldn't use primary cells unless there is any other good reason. And I think now one would say you'd need a very good reason not to be using primary cells, but the amount of vaccine that's out there that's not performed in that way. So it's really about actually enforcing and agreeing what is a reasonable approach to take. And I think, cost comes into it, but cost shouldn't compromise really being able to apply the minimum standard. Because if either the vaccine's ineffective or it's actually dangerous, the fact that you paid half the price for the vaccine, it's not going to pay back. So,
Wannes, did you have something to add to?
Yes. I also want to add that just simple in vitro batch controls can already avoid a lot of damage. If you just do some DIVA assays or some sequence on phylogenetic regions, you would have avoided the disaster with the lumpy vaccine from G-VII from the vaccine. So just simple in vitro batch control, it doesn't have to cost a lot of money. You can avoid a lot of problems.
Thank you. Alejandra?
Yeah, I have a question. No, it's for Amélie regarding the use of comparing the adjuvants, the aqueous and the oil. I understand that all the studies that have been done in the past that were long ago, maybe now the aqueous adjuvants, the allium is different. But I wonder why not only one dose and compare it over time instead of vaccinating several times. And if you have any information in young animals, if the immune response is the same if you use the allium instead of the oil adjuvant. Sorry, 2 of them. One is why you give booster doses. Because the old story is like when you compare the allium and the oil, yes, the oil stays over time with immune response, so the duration is better with one dose than the allium. That's the old story in South America. Although, of course, the allium has changed and everything can be new. And the other one are young animals, if you have any information.
What I said, when we compare, we have never... When I talk only for BI vaccine, we have compared only our Avtovax Pure Avtopor with, we recommend 2 injections after a booster after 6 months, and we have never proved a longer duration of immunity. And maybe in America, yeah. In this case, when we compare, well, as I said, when we compare strictly the adjuvant, it's not only the adjuvant, but also maybe other things, the strains, the strains and the payload also.
Yeah.
That's why it was the purpose of the...
Yeah, this is just one trial.
Yes, but it's not... Yes.
Yeah. And you don't know what's going on in young animals?
Yes, young animals. Yes, just naive animals in Europe, never vaccinated.
Young animals,
with
calves?
Yes, without MDA, etc.
Okay.
Okay, Donald?
Thank you all. There were 5 excellent presentations, and I know it's late in the evening and people are flagging. This is a question that perhaps would most appropriately have been asked of the last panel, and was prompted by a slide that Erwin showed, showing the length of time, the alarming length of time it takes to bring a new product to market. Is there anything we as a community of public and private actors could do to address that? And are there any specific lessons we could learn from the speed with which COVID-19 vaccines were made available? A question for the panel, for all of you, any of you.
I mean, just a couple of observations, Donald. I think what Erwin showed was introducing an entirely new technology. So that, as he said, there were a lot of challenges that they had to overcome during the development. Of course, if you're just introducing another inactivated vaccine, you wouldn't have that kind of development time because everything's clear and it's much easier. I think it's interesting around how did COVID-19 get on so quickly? I hesitate. If it had been a veterinary application, it would have taken several years. It would never have gone through as quickly, and it was just because of the human risk. But I think there are lessons that have been learned from that will apply to humans. From humans to veterinary vaccines, and I think the pathway certainly for RNA vaccines will be much quicker than it would have been had that not happened on the human side.
Yeah, we are also going to discuss all these issues in the GFRA session, so we are planning to do it on the last day.
Did the other panel members have anything to add, or? No, okay. I think Nagendra, did I see your hand? Yep. Nagendra, sorry. Yeah.
Every now and again, I have to ask a controversial question. 25 years ago, when I started my career in FMD, oil vaccines were introduced in India. And the claim made by the companies that had vaccine production facilities then was give 1 dose of oil vaccine, forget for 9 months, give another dose at 9 months, forget for 1 year, and then it becomes an early vaccination. And then slowly the aqueous vaccines, which were performing well, being vaccinated every once in 6 months, was phased out. Today, after 25 years, I'm listening that there is no difference between the aqueous vaccine and the oil vaccine. Why this change? Why is the global south being fooled? Thank you.
Slightly loaded question, Nagu, but that's cool. Amélie?
I don't know if things have changed in 25 years, but maybe the previous study won't compare strictly equivalent parameters, what I explained.
Did you want to mic,
Yeah.
Pascal?
Yes.
You'd like to add something? We love a good heated discussion here, so. Thanks.
Well, if I go back to old history within our company as well, we've always known that it's more difficult to make a good aqueous vaccine than to make a good oily vaccine. So I guess there are different levels of quality between aqueous vaccines. And in the formulation of aluminum hydroxide with the antigen, there are certain steps that you need to follow that make it either equivalent to an oily vaccine or worse. But the oil was also introduced to vaccinate pigs. Huh?
Well, also the alum has changed, I think. So it's not the same product that they were using years ago.
True, the quality of the saponin and the aluminum hydroxide has changed also.
And also the size of the particle that you finally get where the antigen is bound is more good for antigen-presenting cells than it was before. Yeah, that's true.
Okay, we might have one more question. Yep. Oh, okay, yep. Yes, sorry, I don't know your name, I'm sorry. Yes.
Thank you. This is regarding lumpy skin disease vaccine. What I saw in your presentation, what is in vitro safety. Can you elaborate on it?
Sorry, can you repeat it once more?
Yeah, in your presentation, you were mentioning about in vitro safety.
Ah, yeah. Yeah, so in vitro safety is testing a vaccine, checking the strain that is present, so via sequencing of some phylogenetic regions, but also looking at contaminants of, for example, FMD or BTV that can be present, looking at the titer of the vaccine, and if necessary, we can also do whole genome sequencing of a vaccine to have an idea of the safety in vitro of this vaccine.
Thank you. So, Danny, did you have a super quick question with a super quick answer? A comment? Okay.
It's a comment to Donald's question. Of course, foot-and-mouth disease is very complicated because it has 3 serotypes and it's a very complicated vaccine. Just as a comparison, when in 2006, the blue tongue type 8 came out of the blue, a vaccine was developed from literally isolating the strain in the field to putting 80 million doses in the field within 18 months. So it is possible, but then of course it goes with a conditional license and you don't have the whole package. But to do the basics, it's perfectly possible to do it in a short time. But, yeah.
Thank you. I feel like the only way I can keep you guys here is if they start bringing in wine. So I'm going to say thank you to the panel for some very interesting talks. And I just have to fulfill my duties by pointing out to you all that there's a lot of really awesome posters in the poster area. Please go and have a look at them. There is no organised function this evening, but you can still take yourself to look at the posters. And, oh, of course, do not forget to vote for the best poster. I think it closes tomorrow and there'll be a presentation. So thank you so much for your attention and have a good evening. Oh, sorry.
9 o'clock. 9 o'clock tomorrow.
9 o'clock. 9 o'clock start tomorrow. Go for a run. Come in full of energy.